जैसे कि हम ट्रांसफ़र और फिक्सेशन के हवाले से आरएनए की बात कर रहे हैं, हमने बफर्स के हवाले से बात की और जेल की ट्रिमिंग के हवाले से बात की। जब हमारे पास जेल और नायलॉन मेम्ब्रेन, रिक्वायर्ड फ़िल्टर पेपर, बफर, एवरीथिंग इज़ अवेलेबल, देन वी स्टार्ट सेटिंग देम अप स्टेप बाय स्टेप. We are going to talk about the transfer system or the setup of the transfer system. फर्स्ट 6 स्टेप्स की हमने पिछले मॉड्यूल में बात की, जिसमें हमने बात की कैसे ग्लास प्लेट को, कैसे रेजरवायर को, उसके ऊपर एक फिल्टर पेपर को विद ड्रिपिंग edges in the buffer, and then gel is placed upside down. What is next? What we have to follow step by step. At every step there are some points. If they are ignored, then the problem is going to come in the transfer. So once we take the gel and we place it over the plate, which is glass plate, which is covered with the blotting paper, the edges are dripped in the reservoir, then the next thing is to observe if there is any air bubble in between the gel and the filter paper. If there is any air bubble, that air bubble needs to be removed. Otherwise, the transfer from that particular spot will not be possible because liquid will not pass through that part of the gel. The next step would be, अगर हम एक टॉप व्यू लें इस पूरे सेटअप का, this is going to be our tray और the glass baking dish. इसको हमने यहाँ से एक ग्लास प्लेट रखी इसके ऊपर. देन वी हैड आवर फिल्टर पेपर विद द ड्रिपिंग एजेस इनटू द कंटेनर. इसके ये एजेस जो फिल्टर पेपर के हैं ये कंटेनर में डिप्ड हैं. अब अगर देखा जाए हमारे पास ये पर्टिकुलर एरिया है दैट इज़ ओपन, दिस इज़ ओपन, दिस ऑल द 4 sides of the glass baking dish are open, and from this particular point, from this particular open area, evaporation is possible. The buffer can evaporate and it depends on the temperature, ambient temperature. Therefore, in order to make it sure that the transfer of buffer should be only from the gel, we need to cover all these open areas by saran film or by some kind of parafilm that is going to force the buffer, that 20x SSC buffer, just to pass from this particular area that contains the gel. So once we have the gel upside down on the filter paper, this is our lower right corner of the gel. We also cut the nylon membrane as per required size of the gel, but 1 millimeter larger than the gel, and we mark the lower right corner a cut so that we can align the gel and the membrane. So once we place the gel, we are going to place above the gel this nylon membrane. Again, nylon membrane will be made soaked in the relevant or in the required buffers so that it can be shifted over here. अब इस stage के ऊपर again we have to take care of the air bubbles. If there are any air bubble in between the membrane or in between the gel, they are removed. अब यहाँ पे sometimes जो student है वो गलती करता है कि membrane जो है वो properly settle नहीं हुई gel के ऊपर. So once it is placed over the gel, it is not recommended to remove the membrane. Let it be like it and then proceed further. Step number 10 would need 2 filter papers which are cut exactly of the same size of the nylon membrane and soaked in the 0.2x SSC, placed over the nylon membrane, and then again smooth to remove the air bubbles. एयर बबल्स को रिमूव करने का जेंटल तरीका है, आपके पास प्लास्टिक की पाइपेट्स होती हैं 5 ml की या 10 ml की, या फिर ग्लास की पाइपेट्स होती हैं. Gently roll them over so that any air bubble should go off the setup. Then we need some dry paper towels, a stack of dry paper towels which can make a height of 5 to 8 centimeters, that is going to be placed over those blotting papers. which is going to act as absorbent, and these towels are then covered with a glass plate which is going to bear the weight. That weight should be 400 grams up to 1 kilogram. It depends how tall is the tower, so that the capillary action can be started from the reservoir. The concentrated salt solution starts moving through the gel, through the membrane, and through all the paper towels. And while this movement of ionic solution is taking place, RNA from the gel is going to be transferred to the membrane. And this setup can be kept as it is for 4 hours or as optimized by your lab or your protocol. Sometimes this setup may need to be remain in this position overnight. More than 12 hours may be required for this particular setup. Once the transfer time is over, then we are going to dismantle this transfer system, where now the required material is the membrane and the gel. Gel can be processed for staining to see the efficiency of transfer. Membrane is taken and is soaked in 6S SSC buffer for 23 degrees centigrade for 5 minutes with agitation. That is going to be done on a rotary platform, and once this rotary platform step is over, the 5 minutes of agitation is over, the excess of 6S SSC buffer is removed or is dried, and then the membrane is laid down on the blotting paper so as the excess of the liquid can be absorbed. This membrane is now ready to proceed further for cross-linking or for fixing the RNA with the membrane using any of the 3 available methods.